Good afternoon, friends. I Miss Amruta Sem VI today going to conduct a lecture on ICH guidelines. ICH stands for International Conference on Harmonization. So in this slide, today we are going to study the ICH guidelines, quality guidelines that is related with the QSEM, that is quality, efficacy, safety and multidisciplinary, and stability guidelines. Okay, so let us see what is meant by ICH. ICH stands for International Conference on Harmonization of Technical Requirements for Registration of Pharmaceuticals for Human Use. Means whenever a new pharmaceutical product is formulated, it's prepared, first of all it is registered, that is it is safe for the human use. Okay, so for that purpose this ICH have came into existence. ICH brings together the regulatory authorities and experts from the pharmaceutical industries in the Europe, Japan and United States to discuss scientific and technical parameters of the product recognition. ICH is used to avoid duplication of clinical trials in the different countries. Means this ICH is only limited to the Europe, Japan and United States. Okay, means the regulatory authorities, the experts from the pharmaceutical industries of Europe, Japan and United States have came together. They discuss together on the scientific and technical parameters, okay, of the product new product launching, and they come together to avoid the duplication of the clinical trials done in the different countries. Means the same study is done in the Europe, Japan and United States, then it will cause the duplication, it will cause the wastage of the chemicals, death of the animals, death of the human beings. Okay, so to avoid these problems, to avoid the duplication of the clinical trials, these three countries have came together. They have decided the guidelines. They are preparing the draft relating to the launching of the new pharmaceutical product. Okay, so purpose of the ICH is to develop safe, cost effective and high quality medicine, to promote public health, to prevent unnecessary duplication of the clinical trials, to minimize the use of animal testing without compromising safety and effectiveness, to avoid death of the human volunteers, and to import and export drugs in all the three countries. So these are the different purposes for which the ICH came into existence. The ICH members or ICH participant includes, it is comprised of 6 parties, okay, that represent the regulatory bodies and research based industries in Europe, Japan and USA. Means from Europe there is one regulatory body and one research based industry. From Japan one regulatory body and one research based industry, and from USA one regulatory body and one research based industry. In this way, from 3 countries, total 6 parties are the members or the participants of the ICH. From Japan, the Ministry of Health, Labor and Welfare is the regulatory body, and Japan Pharmaceutical Manufacturers Association is the research based industry. So these two are the members of the ICH. From Europe, European Union and European Federation of Pharmaceutical Industries and Association, these both are the participants of the ICH. And from USA, Food and Drug Administration and the Pharmaceutical Research and Manufacturers of America, these two are the participants of the ICH. Additional members include World Health Organization, European Free Trade Association and Canada. The structure of the ICH participants or ICH members is, it consists of ICH Steering Committee, ICH Coordinators, ICH Secretary, ICH Working Group. Under Working Group there are 2. First is Expert Working Group and second is Implementation Working Group. So in this way there is a structure of the ICH committee. Next is the process of harmonization. Process of harmonization means what? That is the preparation of a draft. How the exactly product come into the market. मतलब जो कोई भी प्रोडक्ट नया फॉर्मुलेट करने वाले हैं उसके लिए ड्राफ्ट बनाते हैं. ये ICH के जो मेंबर्स है, 6 मेंबर्स है, दो फ्रॉम 3 different countries. Okay, one is the regulatory agency and another one is the research based industry. All these 6 members from 3 countries come together, they prepare a draft that how the exactly product should come into market, what test it should be performed onto that product, so that that product will be the good for the human health. It will not cause any kind of a harm. It will be of a good quality. So for that purpose, these three countries, the 6 members from these three countries come together and they prepare a draft, and accordingly the work is done so as to avoid the duplication of the work, so as to avoid the death of the many animals as well as the human beings, and to reduce the time for the manufacturing or preparation of a new dosage form. So for that purpose this harmonization is done, and the process of the harmonization starts from the concept paper, that is the summary of the proposal. Okay, here the business plan is also required as per the activity of the harmonization. Any ICH member can give the proposal. Process of harmonization is divided into 4 steps. Okay, mainly the harmonization process is divided into 4 steps. First is the formal ICH procedure, second is question answer procedure, third is revision procedure, and fourth one is the maintenance procedure. मतलब ये 4 पार्ट्स में हार्मोनाइजेशन की प्रोसेस जो होती है वो डिवाइड रहती है. फर्स्ट इज द formal ICH procedure. The steps involved in the formal ICH procedure are: step 1 is a consensus building. In consensus building, what exactly is done? That is the concept paper, means whatever idea is there, new idea, any person, any member of the ICH can or any steering committee member can apply for the concept paper. That is, they can submit the new topic in which they want to they have interest and they want to prepare the new formulation. That is the first step, that is the consensus building. Here draft is prepared on technical document and collected from all the 3 countries. Okay, means whatever study is done onto that particular draft in 3 countries, that all study is collected, that all information regarding that new topic is collected from all 3 countries via email, telephone, conference, web conferences, etc. Okay, and draft is prepared by the steering committee. Okay, then in the step 2a, confirmation of a 6-party unanimity on technical document, means here draft goes for regulatory consultation and is signed by steering committee as a final technical document. Okay, means whatever document is prepared, okay, in the first step, that is sent to the 6-party, that is sent to the steering committee for cross checking. Okay, after that there is a step 2b, that is exposure of a draft guideline. In this step the draft is exposed in front of 3 ICH members. Okay, and this final draft, if they want to make any changes, that ICH members make the necessary changes, and it is signed by the 3 regulatory ICH members from all those 3 countries. Next is the step 3, that is regulatory consultation and discussion. It takes place in 3 stages. First stage is the regional regulatory consultation. In this stage, the draft is discussed with the committee of all the 3 regions, that is Europe, Japan and USA. And name of the regulatory committees are as follows, that is Europe, Committee for the Medicinal Product for Human Use, Japan, Ministry of Health, Labor and Welfare, and USA, Federal Register. And during this discussion, all the regulatory authorities comment on it. Okay, that is the regional regulatory consultation, means there is a discussion related with the that draft in between all these 3 committees. Next point is the stage 2, that is discussion of the regional consultation committee. Means in this stage the review is done on the comments received from the regulatory committee during the first stage, and expert working group works to handle the comment and forms the revised expert draft guidelines. Means actually who is going to perform the work related with that particular new formulation, the expert working group is going to perform the work. So that regional or that regulatory authorities discuss that all draft with the expert working group, and if expert working group have any problem on it, they comment on it. And in the third stage there is the finalization of the step 3 expert draft guidelines. Means whatever comments are given by the expert working group, they are cross-checked, the necessary corrective actions are taken on to them, and the new final draft is prepared and it is forwarded to the steering committee in the stage 3. In step 4, there is acceptance of an ICH harmonized tripartite guidelines. Means here the ICH harmonized tripartite guidelines, that is final draft guidelines, are signed by the expert working group and are accepted and signed by the steering committee. And finally there is a step 4, that is implementation of a guidelines. Means these final guidelines which are signed by the expert working group and by the steering committee are actually implemented. Okay, means in all those 3 countries the work is actually started in the step 5, that is the implementation of the guideline. After that, second main step is the question and answer procedure. Means actually while implementing that guidelines, there are some problems which are faced by the expert working group. They ask that queries to the steering committee and the steering committee gives answer to that questions. Okay, so that is the question and answer procedure. In step 3 there is the revision procedure. Means if the draft tells you that you have to perform this particular work by this way, this procedure you should follow, but if you are not getting the exact result by using that procedure, there is necessary to make the changes. Okay, then that changes are mentioned as revised in that draft. Okay, and that is called as the revision procedure, and they are mentioned as R1, R2, R3, R4, etc. And last one is the maintenance procedure. Means it is done only in case of Q3 guideline, that is alteration of impurities, residual solvents, etc. Means if any sample, if any solvent, if any excipient is not available with us, instead of that we are using the another one. Okay, so that is called as a maintenance procedure. Okay, so these are the different steps of the ICH process of harmonization. Next point we have to study, that is the overview of QSEM. Here all the following technical parameters should be covered while drafting the ICH guidelines. That is, Q is related with quality, S is related with safety, E is efficacy, and M is multidisciplinary. Quality series include following parameters, that is Q1, it deals with stability, Q1A, stability testing of new drug product, Q1B, photostability test of new drug product, Q1C, stability test of new dosage form, Q1D, matrix design for stability test of drug, Q1E, evaluation of stability data, Q1F, package stability data, Q2, analytical validation, Q3 is related with impurities, Q3A, impurity in new drug substance, Q3B, impurity in product, Q3C, guidelines for residual solvent, Q3D, guidelines for elemental impurities, Q4 is related with pharmacopoeia, Q5 is related with quality of biotechnological product like Q5A, viral safety evaluation, Q5B, analysis, Q5C, stability test for biotechnological product, Q5D, characterization of biotechnological product, Q5E, comparison in process change, Q6 is related with specification, Q7, good manufacturing practices, Q8, pharmaceutical development, Q9, quality risk management, Q10, pharmaceutical quality system, Q11, development and manufacturing of the drug substances. Safety includes following parameters. S1 is related with carcinogenicity studies, S2 is related with genotoxicity study, S3 toxicokinetic and pharmacokinetic, S4 toxicity testing, S5 reproductive toxicology, S6 is related with biotechnological product, S7 is related with pharmacology studies, S8 immunotoxicology study, S9 non-clinical evaluation for anti-cancer pharmaceuticals, and S10 photosafety evaluation. Efficacy includes E1 and E2 clinical safety, E3 clinical safety report, E4 dose response studies, E5 ethnic factors, E6 good clinical practices, E7 to E11 related with clinical trial, E12 is related with guidelines for clinical evaluation by therapeutic category, E14 is related with clinical evaluation, and E15 and 16 are related with pharmacogenomics. Last one is the multidisciplinary, includes M1 med-DRA technology, M2 is electronic standard, M3 is non-clinical safety studies, M4 is common technical documents, that is CTD, M5 is related with data element standards for drug dictionary, M6 is related with gene therapy, M7 is genotoxic impurity, and M8 is ECT, that is the electronic common technical document. So these are the QSEM, that is quality related guidelines of ICH. And last point is the ICH stability testing guidelines. The following guidelines denotes the stability testing requirement for registration of a drug or drug products in only ICH countries. Means these guidelines are only related with the ICH countries, that is Europe, Japan and USA, and they are not suitable for import or export of the drugs in other countries in the world. Okay, so the objective is, the main purpose of stability testing is to provide the evidence on how the quality of active ingredient or drug product varies according to the change in different environmental conditions like temperature, humidity, light, etc. Means in case of stability testing, we are checking that product is suitable at what temperature. Okay, for the mentioning the storage condition onto that label of that product, the stability of the product is tested on different areas, on different temperature, on different humidity areas. Okay, another purpose of stability testing is to establish the storage condition, retest period, and shelf life of the drug and a drug product. Means another reason why we are performing the stability testing is to decide the storage condition, to determine the retest period, means till how much time this particular product is stable and at what time it should be again tested, and to determine the shelf life of the product, that is the expiry date of the product. The different definitions related with the stability testing are: first is accelerated testing. Here the rate of chemical degradation or physical change of an active drug substance or drug product is increased by using exaggerated storage condition. Accelerated means we are forcefully using the exaggerated storage condition, that is by varying the temperature, humidity, and other storage condition, by forcing the substance at a high temperature, at more humidity. Okay, at exaggerated storage condition, we are checking what effect is happened on that particular product, how much the chemical degradation or physical change has been occurred in that product. That kind of a testing is called as accelerated testing. Second is drug substance. The unformulated drug which may be formulated by adding excipients to produce the drug product. Means drug substance means active ingredient which is added with excipients, and after that we get the drug product. Climatic zone. The whole world is divided into 4 zones depending upon climatic conditions, and that is called as a climatic zones. Excipient means anything other than the active drug substance in the dosage form is called as a excipient. Example, in case of paracetamol tablet, paracetamol is active drug, and anything other than the paracetamol like the talc, magnesium stearate, etc., any binder, etc., they act as a excipient. Next is the drug product, means drug substance along with the excipient, means active drug, active ingredient plus excipient. The final immediate package ready for marketing is called as a drug product. Next term is the expiry date. The date mentioned on the label of formulation which indicate the period when product remain within the shelf life specification if stored under defined condition and which is not safe to use afterwards is called as expiry date. Means till that date that product is stable, it is as per the specification, it matches to the standards. Okay, and after that if we use it, it will produce the harmful effect. That period is called as, that date is called as the expiry date. Next is the formal or systematic studies. The studies undertaken to a pre-approval stability protocol which embrace the requirement of these guidelines are called as formal studies. Long-term or real-time testing means evaluation of a physical, chemical, biological, microbiological characteristic of a drug and drug product up to the shelf life and retest period is called as a long-term testing. म्हणजे त्याची शेल्फ लाईफ किंवा त्याची रिटेस्ट पिरियड येईपर्यंत आपण त्याची टेस्ट कंटिन्युअसली त्याला मधून त्याचे फिजिकल, केमिकल, बायोलॉजिकल, मायक्रोबायोलॉजिकल कॅरेक्टरिस्टिक चेक करत राहणे. दॅट प्रोसेस इज कॉल्ड एज अ long-term testing. Next is mass balance or material balance, the process of addition of assay value and degradation value and see how it add up to 100% with due consideration of margin of analytical precision. म्हणजे बॅलन्स करून बघतो आपण की असेच व्हॅल्यू किती आला आहे, म्हणजे आता किती रिझल्ट आला आहे, डिग्रेड किती झाला आहे, मग त्या दोघांची सम मिळून जेवढं आपण ॲक्च्युली घेतलेलं ते 100% तेवढ्याला मॅच होतं आहे की नाही आहे. त्याच्यामध्ये थोडे तुमचे जे काय ॲनालिटिकल म्हणजे वेईंग करताना थोडा वेस्टेज झाला असेल. ओके, सो दॅट वी do the minus in it. Okay, but the addition of the assay value and the degradation value and some error of the margin should be equal to 100% whatever we have taken. Okay, that is called as a mass balance or material balance. Next is the matrixing, the process of randomly selecting the fraction of a product from whole batch and testing them at any sampling point is called as a matrixing. म्हणजे बॅचमधनं मधून कोणताही प्रोडक्ट आपण रँडमली सिलेक्ट करायचा आणि त्याच्या आपण टेस्ट परफॉर्म करायच्या. दॅट प्रोसेस ऑफ सॅम्पलिंग okay is called as a matrixing. The product tested represents the whole batch. Sampling should be done in order to check uniformity in the strength, container size, different batches, container and closure system etc. Means या सगळ्या वेगवेगळ्या प्रकारच्या टेस्ट तुम्ही मॅट्रिक्सिंग करून चेक केल्या पाहिजेत की कंटेंट युनिफॉर्मिटी आहे का, कंटेनरची साईज व्यवस्थित आहे का, बॅचेस सगळ्या व्यवस्थित मॅन्युफॅक्चर झाल्यात का, कंटेनर क्लोजर व्यवस्थित सुटेबल आहेत की नाही आहेत. ओके, सो मॅट्रिक्सिंग केलं पाहिजे. अँड दॅट मॅट्रिक्सिंग, that sample we are using, it represents the whole batch. Next is mean kinetic temperature. It is higher than the mean arithmetic temperature and calculated by the Highness formula. Next is retest date. The date when sample of a drug substance should be re-examined to ensure that material is still suitable for use is called as a retest date. म्हणजे पुन्हा कधी आपण आज टेस्ट केली पण ह्याच्यानंतर कधी टेस्ट करायची की चेक करण्यासाठी की त्याच्यानंतर तो पर्टिक्युलर प्रोडक्ट स्टेबल आहे की नाही आहे. ओके, दॅट date of retesting is called as a retest date. Next is retest period. The duration in which the drug and drug product remains within the standard specification if stored under defined condition is called as a retest period. म्हणजे असा ड्युरेशन, अशी वेळ की जोपर्यंत तो प्रोडक्ट तुमचा आहे या स्टँडर्ड स्पेसिफिकेशन म्हणजे त्याची स्टॅबिलिटी आहे अशी राहते, तो डिग्रेड होत नाही आहे, त्याची स्टॅबिलिटी लूज होत नाही आहे. ओके, त्या पिरियडपर्यंत दॅट पिरियड इज कॉल्ड ॲज अ रिटेस्ट पिरियड. Next is primary stability data. The stability data obtained after the storage of the drug and drug product is in defined packaging material under prescribed storage condition which support the proposed retest date is called as a primary stability data. म्हणजे तुम्ही स्टोअर करून ठेवला प्रोडक्ट आणि त्याच्यानंतर आपल्याला त्याच्या रिटेस्ट पिरियडपर्यंत आणि त्याच्यामध्ये आपल्याला जो काही त्याचा डाटा मिळाला की त्याची स्टॅबिलिटी लूज झाली आहे नाही झाली आहे, पॅकेजिंग मटेरियल बरोबर सुटेबल आहे नाही आहे, दॅट डाटा इज कॉल्ड ॲज अ प्रायमरी स्टॅबिलिटी डाटा. And shelf life means the period when drug product remain within the approved shelf life specification under prescribed storage condition in proposed container and closure is called as a shelf life. म्हणजे आपण डिझाईन केलेलं कंटेनर आणि क्लोजर म्हणजे त्याच कंटेनरमध्ये त्या क्लोजरमध्ये तो पर्टिक्युलर प्रोडक्ट किती दिवसासाठी आपण स्टोअर केला तर स्टँडर्ड स्पेसिफिकेशनमध्ये त्याचे रिझल्ट्स दाखवतो आहे, म्हणजे तो डिग्रेड होत नाही आहे, स्टेबल राहतो आहे. दॅट टाईम इज कॉल्ड दॅट time span is called as the shelf life of that drug. Next is the elements of stability testing. While stability testing, we have to do stress testing, means it helps to determine the intrinsic stability of a molecule. Here product is expired to the degradation condition to identify the degradation product. As per name suggests, stress testing म्हणजे स्ट्रेस आपण द्यायचा, मीन्स आपण खूप जास्त डिग्रेड होईल अशा कंडिशनला त्या प्रोडक्टला एक्सपोज करायचं आणि बघायचं की त्याचं डिग्रेडेशन प्रोडक्ट काय आहे, तो किती हार्मफुल आहे. ओके, दॅट इज द स्ट्रेस टेस्टिंग. सेकंड इज सिलेक्शन ऑफ बॅचेस, मीन्स ॲटलीस्ट 3 batches should be selected for stability testing. Accelerated and long term testing should be done. Test procedure and test criteria. Stability testing should cover physical, chemical, microbiological testing and validated methods must be applied. म्हणजे IP, BP, USP त्याच्यामध्ये मेन्शन केलेल्या व्हॅलिडेटेड मेथड्स प्रोसिजर्स अप्लाय करूनच तुम्ही स्टॅबिलिटी टेस्टिंग परफॉर्म केलं पाहिजे आणि त्यामध्ये फिजिकल, केमिकल, मायक्रोबायोलॉजिकल अशा सगळ्या प्रकारच्या टेस्ट तुम्ही परफॉर्म केल्या पाहिजेत. Next is the storage condition. There are 2 storage conditions should be tested of every product. Long term testing साठी 25 degree Celsius plus minus 2 degree Celsius at 60 plus minus 5% relative humidity for 12 months. आणि accelerated testing साठी सगळं जास्त, टेम्परेचर जास्त, ह्युमिडिटी जास्त पण टाईम कमी, त्यात आपण ते बघतो की तो प्रोडक्ट तुमचा डिग्रेड झाला आहे का स्टेबल राहिला आहे. सो फॉर ॲक्सेलरेटेड टेस्टिंग टेम्परेचर इज 40 plus minus 2 degree Celsius at 75 plus minus 5% relative humidity for 6 months. Next is testing frequency. In long term testing should be done after every 3 months in first year and every 6 months in second year and annually in the third year. म्हणजे पहिल्या वर्षी प्रत्येक 3 महिन्याला टेस्टिंग केलं पाहिजे, दुसऱ्या वर्षी प्रत्येक 6 महिन्याला आणि तिसऱ्या वर्षी वर्षातून एकदा असं तुम्ही प्रोडक्ट स्टॅबिलिटी टेस्ट केली पाहिजे. नेक्स्ट, packaging or container used in long term testing should be same as that of the actual packaging material used for storage and distribution. Evaluation, it should not only cover the assay value but also level of the degradation product. आणि लेबलिंग specific requirements should be mentioned on the label regarding stability of a drug, and terms such as ambient temperature, room temperature are unacceptable. Directly temperature range जी आहे ती मेन्शन केली पाहिजे. ambient temperature, room temperature असे शब्द तिथे लेबलवरती नाहीत मेन्शन केले पाहिजे. So these are the elements followed into the stability testing. Here we have completed with the point, that is ICH guideline. Thank you.